Hello everyone, in this video we are going to talk about a genetic switch, which is a mechanism takes place in lambda phages, which are bacteria phages. These are simply viruses that affect bacteria. They infect bacteria and multiply in them. Now scientists earlier working on them observe that they can easily switch between their two states, one being a dormant state and the other being a active state. Now in dormant state they don't harm the bacteria, they undergo lysogeny cycle, and in the active state they lyse open the bacteria to produce hundreds of progeny phages. Now this biological process is based on functioning of several genes. They're switching on and off, and the regulation of these genes at various levels, which we'll see in this, we will go through step by step in this video. Now we shall start with the structure of lambda phage. So this slide shows the structure of a lambda phage, which is observed under a microscope. This is a head, collar, tail fibers. Now the genome of a lambda phage is constituted by a single DNA molecule which is wrapped in a protein coat and present in the head. Now when phage is infecting E. coli cell, it attaches to the surface with the help of these tail fibers, and then it drills a hole in the cell wall and squirts its chromosome into the bacterium, leaving its coat behind. Now when the lambda DNA is inside the bacterium, it can choose two fates. One fate being a lytic cycle, where it enters where some genes are turned on and off according to a precisely regulated program, which allows its replication and production of new head and tail proteins, and leading to synthesis of new phage particles, which kills the bacteria cell open and then infect other new bacteria cells. The other fate being that some different phage genes are turned on and off, and the one phage chromosome becomes the part of the host chromosome called as a prophase. Now this prophase grows and divides with the bacteria without disturbing the progeny bacteria. It gets inherited in the progeny bacteria also without killing the cell. Now there are some inducers like ultraviolet light, which can allow this prophase to enter a lytic cycle, leaving behind its lysogenic cycle. So now we're going to explore what are the genes that help the lambda phage to enter the second state, that is the lytic state, from the lysogenic state. A simplified map of lambda chromosome is shown in figure. Now, as you can see, only 6 genes are named individually in the representation: Cl, C2, C3, N, Cro, and Q. There are 2 genes present in the DNA replication region, which are not shown. There are about 3 genes present in the lysis region, and a recombination region hosts about 10 genes. The head and tail regions codes for the genes required for lambda phage packaging. The cos site here is used in circularization of phage DNA molecules, and at site is the site for integration into the host chromosome. As we know, in a lysogen, the single phage gene that is getting expressed, which encodes for a lambda repressor. This protein acts as a both positive and a negative regulator of a gene expression. It binds to 2 operators on the lambda DNA, as it turns off all the other phage genes, as it turns on its own gene. Now we know that under ultraviolet radiation the lysogens switch to lytic cycle by inactivating repressor proteins, but a second fast regulatory protein CRO is synthesized. This CRO, which promotes and is essential for the lytic growth to take place, also binds to DNA at the same two sites but with opposite physiological effects. Now the gene coding for this repressor and CRO protein are named as CL and CRO. These are present adjacent on the lambda chromosome, but they transcribe in opposite direction. The start points of these transcriptions of these two genes share a common region where each of the promoters are present. The PRM, which is the promoter of repressor maintenance, as the name suggests, is the promoter for CL gene, which is going to code for our lambda repressor protein, and the PR, which is the right promoter, which is the promoter for the CRO gene. Now these promoters, in between these promoters there are three operator sites which have overlapping region in between them, which will play a major role in the switching over of these lysogenic to lytic cycle. So the operators are OR1, OR2 and OR3. Now there is also another key point which we should remember, that when RNA polymerase binds to either of the promoter sites, see, the RNA polymerase is going to bind to the promoter sites. Now when it binds to PR, it does not need any help, whereas when it has to bind to PRM protein PR promoter, it needs help of an activator protein, which is none other than the repressor protein itself. So this is what I said in the starting of this slide, that the lambda repressor turn on its own expression as well. This is how it does that. We're going to talk about it in more detail in the next slide. We're also going to see at what levels does these expressions taking place, and at what levels the genetic switch is getting affected, the transcription level, post transcription level, translation level, and even at the protein stability level. Now the positive control of PRM by lambda repressor protein, as we can see in the figure, is when it binds to the OR2 operator site, enabling the polymerase to sit at PRM and begin transcription. This is the positive regulation which increases transcription of its own gene. Now the negative control can be when it binds to the operator 3 and it does not allow RNA polymerase to bind to PRM, and hence as the RNA polymerase requires repressor protein activation to transcribe CL gene, but in for the CRO protein it does not require the same, and hence it can sit on the operator 1 site and start transcribing promoter PR. So next in order is the negative control of PRM and PR promoters by CRO protein. Now the CRO protein, as we know, is synthesized when an induction like the lambda repressor protein is inhibited and an induction towards lytic phase has initiated. So the CRO protein, which binds to operator region 3, suppresses the further synthesis of repressor as polymerase cannot bind to PRM. Now as the lambda repressor's concentration is going down, because an inducer effect is going on, hence the operator 1 site is vacant for the RNA polymerase to bind. So it binds and the CRO production is henceforth initiated. So it acts as a negative control of PRM by binding to the operator 3 side and not allowing RNA polymerase to bind further. When there is sufficient level of CRO protein in the bacteria, the increasing concentration as the crude protein has same affinities. It though binds to operator 3 initially, but it has the same affinity to all the three sides of the operator region. Hence, when a substantial amount of concentration has increased in the cell, the crude protein further binds to the operator region 2 and as well operator region 1, down regulating its own synthesis, which acts as a negative control of its own promoter PR. Now we know lambda repressor has greater affinity or highest affinity for operator region 1, and hence it binds to that region, which does not allow RNA polymerase to bind to PR promoter and synthesize chrome protein, hence negatively regulating PR promoter. Now there is a presence of PL promoter which synthesizes an N protein, which is essential for the lytic cycle. So as the lambda repressor is on and it is needed for lysogenic cycle, so eventually the PL promoter should be switched off. Now it is regulated as when lambda repressor is present, PL promoter produces a short RNA that is of N protein, and it is not able to produce a complete N protein, and hence it really controls the gene regulation of PL promoter as well. As earlier in this video I told you that in order for phages to undergo a lysogenic cycle, lambda repressor protein needs to be present, which switch off all the other genes except its own. Now you might ask me where that lambda repressor came from. So when the phages were undergoing lytic cycle, group protein was getting synthesized and further downstream genes were also getting transcribed. One of the downstream gene was a C2 gene, which produces a C2 protein which acts on two kinds of promoters which helps establish lysogenic cycle. One of the promoters is PRE. RE stands for repressor establishment. Now the PRE promoter produces this lambda repressor protein which will be needed to act on the PRM, which will help bind RNA polymerase, and hence the whole switching of lytic into lysogenic cycle takes place. Now the other role of C2 protein is getting bind to PI promoter, which is the integration promoter which helps phages to integrate into the host chromosome. Now when PRE is producing lambda repressor, it gets bind to operator region 1 and down regulates and group protein synthesis, which further down regulates Q protein synthesis as N protein is not available, and hence this C2 protein controls positively PRA and PI. ND domination by N and Q proteins. The N and Q proteins regulate gene expression at transcription level by preventing termination of transcripts. Now the N protein is seen in action during early phase of infection, where it acts upon NUT N utilization sequences and allow transcription to continue in genes left to N gene and right to CRO gene by preventing termination of these transcripts due to DNA signals. Now the Q protein works in late lytic cycle via the same mechanism as N protein. It anti-terminates a small RNA beginning from, you can see in this figure, PR prime promoter located right to Q when anti-terminated by Q protein. Here you can see the Q protein. This mRNA extends to the head and tail regions. These are the head and tail regions with genes, which will code for head and tail proteins, which help in packaging, allowing lytic cycle to proceed further. During establishment of lytic cycle, Genes to the right of N gene are transcribed in early lytic stage, including genes IND, cis, XIS, and CYP. They are transcribed into a same RNA molecule. Now, for lytic growth, a high quantity of cis is required in comparison to IND. So, a retroregulation mechanism has been evolved. As you can see, CYP is present at the end of RNA. Now, CYP bearing RNA forms a hairpin loop structure which attracts RNase 3. Now, RNase 3 would be present in abundance as the phages are undergoing lytic cycle, as the bacteria are present in rich media. This is all interlinked. Now, this attracts RNase 3, it is cleaved, which causes sequential degradation of IND, hence resulting in high relative amount of cis. As we know, N protein synthesis was getting inhibited by the presence of Lambda repressor protein. This was mainly caused by the production of stem loop structures at the Shine-Dalgarro sequences, and hence preventing the further translation. Now, when the cells are present in rich media, phages usually undergo lytic cycle, because in rich media there are large abundance of N protein and there are abundance of RNase 3 protein, which cleaves these stem loop structures and hence enables the translation to move further. Now, the N protein also binds to N utilizing sequences, NUT sequences, which is present on either side of the CL gene, left and right, which interacts with RNA polymerase and helps juggernaut and surpass termination sequences and produce proteins which are helpful in lytic cycle, that is recombination genes and C3 genes. Their productions are enhanced. C2 protein is essential for the cell to undergo a lysogeny cycle. Hence, the stability of C2 protein acts as a junction where the lytic and lysogenic pathways diverge. Now, the stability of this protein is affected by environmental factors. For example, if E. coli cells are growing in rich medium, we have plenty of abundant active proteases which will further degrade C2 proteins. Hence, no repressor is synthesized and there is abundance of Q and CRO proteins which will lead the cell to undergo lytic growth. Now, if the cells are growing in poor media, they lack active proteases, hence there will be a buildup of repressor proteins which will not allow other proteins to get synthesized, and the bacteria will undergo a lysogeny cycle. Now, there is also present a protein called as C3 which protects C2 and helps in establishing lysogeny. Now, you might remember from the starting of the video that there was an induction by UV light which caused this switch from lysogeny to lytic cycle. Now, how does it really occurs in the bacterial cell? What is actually taking place which causes this switch? Now, in presence of UV, a RIC-A protein is activated, which is a protease and is there to protect the DNA from damage, which could be caused by UV light. Now, this protease cleaves the repressor between amino and carboxy terminal. This cleavage results in loss of effective binding of repressor on promoters. Hence, as repressor is cleaved, this cell gets induced into lytic cycle, as CRO proteins would be produced and further other proteins which are necessary for head and tail, and new phages would be packed inside the bacterial cell. Thank you for hearing me out.